Great, thank you, Janos. Well, thanks so much for the invitation to come here and talk to you guys a little bit about our interest in the area of the human microbiome. And I should say that although most of our work to date is in human, I'm really interested to meet up with folks who are interested in more agriculturally relevant microbiomes. That's pretty high on our priority list of where we'd like to go next with a lot of this research. So, like Janos said, I've been at MIT since 2006. We work in a lot of different areas, and the human microbiome is actually something that we've come into somewhat recently. And one of the, so a lot of my background is in microbial ecology and more environmental microbiology. One of the things that we did in the early days, of course, and you know, I participated in this vigorously as did most folks in the field, is just describing, right? You know, can we make pie charts and things like this of which species of bacteria are in which environments. And now we're getting to a stage where we want to move beyond those pie charts and we want to start understanding, you know, what's the, what are the functional activities going on in the microbiome. And most importantly to me is we want to engineer the microbiome, okay? And one of the points that I like to make is that we're already engineering the microbiome. So whether we're talking about antibiotics, certainly something that probably everybody in this room has used to engineer their microbiome. We still don't really have a good sense of what the long-term impacts of that engineering intervention is. Probiotics, probably everyone is also taking part of the probiotics. And more recently, we're starting to use fecal microbiota transplants where we say, you know, if someone comes down with a Clostridium difficile infection and it's recalcitrant to antibiotic therapy, then we know that we can sort of get that gut ecosystem up and running again just by putting bacteria from a healthy gut into that patient. And this therapy now has about a 95% cure rate for C. difficile infections that have failed antibiotic therapy at least 3 times, sometimes many more. Okay. So we're just at the stage where we're starting to have these very crude tools to engineer the microbiome. So in the first part of my talk, I'm going to try to make this argument to you about the importance of the microbiome in 3 different points. One is that the microbiome reflects the health status of the host. And so I'll show you how we're using the information we get about the microbiome to learn something about the host, in this case, diagnose a disease. I'll show you that diet and environment and behavior can affect the microbiome, okay? And then finally, I'll close the loop and show that by changing the microbiome, we can actually impact the health status of the host. Okay. All right, so in the first story, this is work done in collaboration with Athos Bousvaros over at Children's Hospital. He runs a pediatric inflammatory bowel disease center, and a really fantastic MD PhD in my lab, Ailey Papa. And this started when Athos came to me and said, Eric, you know, look, we've got a problem. Inflammatory bowel disease, not in an adult, and someone like me, you know, it's fairly easy to diagnose. But let's say in an 8-year-old kid, it can be very difficult to diagnose. And at the time that we started the study, there was about a 3-year lag time between the actual onset of disease and the first correct diagnosis. And what that means oftentimes is that the first intervention is surgical, whereas if they had caught and diagnosed earlier, they could have controlled the disease with meds. And so that certainly is a bad outcome for that child. On the other hand, most of the kids that are referred to the IBD clinic don't have inflammatory disease. They have some sort of GI disease, but it's not inflammatory disease. So there's a trade-off. You don't want to send every kid who comes in the door out for a colonoscopy. Okay, that's really, you know, nobody's going to bring their 8-year-old kid to a clinic that is guaranteed to send them all out for a colonoscopy just in case. But on the other hand, you really, really don't want to miss the diagnosis, okay? Because these kids come in with symptoms like weight loss and depression. They often get sent off to the hospital psychologist, and it's like, you know, years later, and then they're on the fast track to colon resection. Okay. So there have been some papers up to this point that said there's a link between the microbes in our guts and health status with respect to inflammatory bowel disease. And there were a number of reasons why this just because someone had seen a correlation, it might not work. Okay. First, the test was not very sensitive at all. Okay. It was very specific, but it was not very sensitive, which is what you need in a pre-screen to see who you're going to perform a much more specific colonoscopy on. Secondly, it was done in adults. And third, the previous study, for their control group, they used terminal cancer patients. Okay, so it was very unclear. And I should say, fourth, they used tissue biopsies instead of stool. Okay, and of course, if you're doing a tissue biopsy of the colon, you've already got your scope up there, so that's not helping us at all. We wanted to see if it would work in stool. So we changed all these variables. And we collected, in the first round, we collected about 105 patients. Okay? What you can see here, these are our controls. So these aren't healthy kids. These are very sick kids who were referred to the clinic. Okay? But after further evaluation, our clinical partners decided they didn't have IBD. They had some other GI illness. These are the two flavors of inflammatory bowel disease: Crohn's disease over here and ulcerative colitis over here. Okay? So these are our patients divided into these three columns. Okay? And the rows are all the different bacterial groups. And now, if it's a darker shade of blue, that means there's more of that bacterial species in that particular patient. So the first thing I want you to notice is that there are no good classical biomarkers of disease here. There's no one species you can point to that would say, aha, if you've got, let's say, E. coli, for example, you've definitely got inflammatory bowel disease if you have large amounts of E. coli. That's true, but using that strategy, you would only correctly diagnose maybe 15% of the kids that came in the clinic. So that is not a very sensitive test. Okay? Instead, what we have is a lot of very weak predictors. Okay? So, for example, if you have a lot of these alley steeps, then you're probably more likely to be in the control group. Okay? But there's still plenty of ulcerative colitis and Crohn's patients who do have a lot of that species. And the same holds true for most of these sort of usual suspects that we expect to find in the gut. Okay? And it was by applying some here, now we bring our clinical network together with our computational background, and we took all of these very weak biomarkers and combined them using a machine learning method. Initially, we used support vector machines, but we found that for this type of problem, random forests seem to work about as well as any other method. And since it's less tunable parameters, that's sort of our go-to method right now. And we actually got some very nice predictions, and we could very accurately predict health versus disease. And it turns out that this non-invasive stool-based test is about as accurate as the leading clinical test, which is fecal calprotectin ELISA assay. And the nice thing about our method is the more patients that we diagnose, the better the model gets, the better the prediction gets, and we can tune that sensitivity specificity dial wherever we want it to be based on what the clinicians need. So hopefully I've convinced you that the microbiome itself can be used to reflect the health status of the host, and we use that to develop a medical diagnostic for inflammatory bowel disease. The next story I want to tell you about is how the environment, okay, our behavior, our diet, our lifestyle affects the microbiome. So this is a study that my graduate student really wanted to do. And what we wanted to do is enroll a bunch of people, measure absolutely everything about their lives for a year. Okay, we wanted to know, when they went to sleep, if they got up in the middle of the night, we wanted to know it. If they traveled, we needed to know that. If their mood changed, okay, if they felt thirsty, if they, every morsel of food that went in their body, we wanted them to record it, record the time they ate it, okay, record how much of it they ate. And of course, every time they had a bowel movement, we needed a sample, okay, so we could go back and sequence it. Unfortunately, nobody signed up for the study. Okay. So Lawrence and I took it upon ourselves to enter ourselves in this in the study. And we really recorded over 300 different aspects of our day-to-day life for a year. Okay, Lawrence said, "Well, gosh, that's a lot of information. If I customize an iPhone app to collect all that information, can I charge an iPhone to the grant?" I said, "Sure, go ahead." So now, if you're interested, there's an app for that. Okay, and this is what we saw. So this is my microbiome on the top. This is my microbiome in 2009. On the bottom is Lawrence's microbiome in 2009. Okay, and this is a lot of data, but what I want you guys to take away from it is that Lawrence's microbiome and my microbiome are very distinct. And although there's variation day to day, week to week, month to month, year to year, in both of our microbiomes, that variation doesn't mask whose microbiome you're looking at. So to put it another way, I could take any of the samples from my microbiome and I could show it to you and you would instantly know that it came from me and not from Lawrence, right? The other thing I want you guys to notice is that the day-to-day variation is about the same as the week-to-week variation, is about the same as the month-to-month and year-to-year. So we have this, this variation within a person on a very rapid time scale. But overall, there's a stability. Okay, there's day-to-day variation, but the overall makeup of the microbiome is very stable over time. Okay, so for the computer science folks, more mathematically inclined, we can get more specific and quantitative about what we mean by stable. Okay, so most of the bacteria we look at, if we plot their abundance over time, it really looks like it's being drawn from a stationary distribution. So there can be some autocorrelation from one day to the next, but over long periods of time, like a year, it really looks like if these are being randomly sampled from some distribution at the beginning of the study, they're being sampled from the same distribution with the same mean and the same variance and everything identical at the end of the study. Okay, so there's some method that allows variation, but holds the average point stable over time. We can look at a few cases where we observe species that are not stationary over time. And one thing we notice is that they tend to cluster together. So this is the tree, a tree of life of the species that are growing in, I think, Lawrence's gut. Okay, and what you notice is the red indicates species that reject this hypothesis of stationarity over time. And they tend to cluster together in groups. And when we look into one of those groups, we see, well, here's this black species here. It appears to be stationary. And then one day it completely plummets. And so this is a log scale, so it completely plummets. It basically gets wiped out. But on the same day, this other gray species jumps up and sort of takes over where the black one left off. So we see that when we observe these non-stationary traces, they correspond to things that make sense ecologically. So one competitor is coming in and then excluding a previous member from that niche. There's a takeover of a niche. Well, what surprised us, of course we expect this happens, is that it could happen so rapidly. It could happen in a single day. Okay, so we recorded all of this information. And, I recorded how much time I spent recording information. and it was about 8 hours a week. So that's like a whole extra workday lost, you know, just thumbing stuff into my iPhone or driving to the office because I had to have a bowel movement. Okay, and so we really hoped that all of that effort would pay off. Okay, so out of all these things we tracked, what are the things that actually correlated with the microbes? And what we turned out is that diet was a very important factor. And what you can see here, this is how much fiber Lawrence eats on a particular day. And this is a group of 4 different bacteria. Okay, they're not phylogenetically necessarily related to each other, but they're related in that as a cluster, they all respond to fiber. Okay, and so if you add up the abundances of those guys, they can account for up to half of the bacterial mass in Lawrence's gut, or at least in his stool. Okay, and it really correlates very nicely in a linear way with how much fiber he eats the day before. So here's the complete list of all of the different correlations that we found. Okay, and time series analysis, especially when things are autocorrelated, can get pretty tricky. Okay, so after doing a lot of statistics to try to tease out these correlations, we were very happy to see that the correlations always went in a direction that made sense. Okay, the bacteria that are in your gut today don't affect the food you're going to eat tomorrow. Okay, that makes sense. But you know, after you do enough statistical backflips to analyze these types of data, you're not guaranteed to have sensible results like that. So it's always the diet and the things you do today that affect the bacteria tomorrow. And the other interesting thing was they don't affect the bacteria that are in your stool today, because presumably that you know, that's been in there for a while, and it depends on what you did the day before. Okay, so the one exception to that rule was stool hardness was definitely affected by the bacteria on the same day. Okay, yeah, we went there. Yeah. So a lot of these associations are basically related to fiber. Okay, there were a few interesting ones. Yogurt was related to bifidobacteria, which are commonly used to produce yogurt, so that made a lot of sense. We found some interesting correlations between citrus and F. brusnitzii. So F. brusnitzii has been found to be associated with protection from inflammatory bowel disease. And so it's also known to in the laboratory to grow with pectin as a sole carbon source. So there's a lot of pectin in citrus rinds and things like that. So this could be potentially a strategy for, as almost a prophylactic for inflammatory bowel disease, eating enough pectin to raise your F. brusnitzii. Your dental hygienist was right. Okay, flossing is negatively correlated with the amount of strep mutans, the bug that causes dental caries. But generally, I would say most of the things we saw had to do with fiber. Okay, there were a few bugs that were inexplicably associated negatively with soup. No idea what that is. Okay, so we also had some, you know, so the data that I'm showing you, you know, first I said, look, there's this day-to-day variation, but overall things are very stable, right? And so what I mean by that is generally the same species are in your gut, and the amount of them that you see on any given day depends on all of these dietary and behavioral traits. Okay, but then you generally don't change which species are in your guts, and that's what really separated me from Lawrence, for example. So now I'll show you some bigger picture stuff about the sort of the entire community. On the previous plot, what I showed you, where everything looked very, very similar, you're only looking at the most abundant things because I weighted them by how abundant they are, of course, right? Here, what I've done is I'm showing you all the different bacteria on the rows of this plot, okay? And then they're all relative to their median abundance, okay, on the columns here. So this is time, and these are bacteria. And so the redder colors means there's more of that bacteria on that particular day, and the blue colors means there's less of that particular bacteria on that particular day. Okay. So what happened, we started the study. A few months into it, Lawrence said, oh, you know, his girlfriend, now his wife, had got this really fantastic opportunity. She was in med school at the time to do an intern in Bangkok. He's like, can I go and hang out in Bangkok over the summer with her while she does this intern? I said, sure, but you have to stay on the study, okay? So we shipped him off a little mini fridge for all his samples, okay, much to his wife's delight. So he could store those while he was in Thailand, and that turned out to be a very good idea because this is the time when he got to Thailand. So you can see everything is going great, and then all of these bacteria suddenly disappear from his gut, and they get replaced with this cocktail of 3 different enteric pathogens almost as soon as he gets off the plane. These pathogens stayed with him during his entire visit to Thailand, okay? And then as soon as he got back to the US, his microbes went back to normal. Okay? So you can get these very severe perturbations of the microbiome, and then when the environmental pressure is lifted, things can snap back to normal. When I was on the study, a good friend of mine invited me out for brunch. I made the mistake of ordering the chef's special French toast, okay? And 48 hours later, I was deep in the throes of a terrible salmonella infection, okay? I went to the urgent care. He said, it's a virus, you don't need antibiotics. But now, doc, I have DNA evidence. It was definitely food poisoning. So as soon as I got the salmonella, you see the same thing happens to me. All of these species disappear, okay? Some of them came back, but actually the majority of the species that disappeared after the salmonella infection never came back. And they were replaced with these bacteria over here. And if we now look on the tree of life, what I'm showing you in the very outer ring, the blue are the species that disappeared. The orange are the species that came in to replace them. And you see all of these species are being replaced by very similar species. It's not the exact same strain, but my ecosystem was repopulating itself with exactly the type of species that it was used to. Not exactly the same species, but very, very similar. Okay? And so somehow, when I showed you those graphs of me and Lawrence, and I said these are very, very different sets of species, that was maintained pre and post salmonella infection, and that's why on the original plot you didn't even see it, because you know little shades of purple were being replaced by other shades of purple, and overall it looks very, very similar. And they were maintained in about the same abundance before and after. And this is something that we think is really, really interesting. We don't have a good theory to explain why everyone's set point for all of these different bugs is different and what keeps it stable over time. So this is what I think is one of the more exciting areas for future research. Okay, so far I've told you that the microbiome can reflect the health status of the host. We use that to develop a medical diagnostic. I've shown you that behavior, diet, lifestyle can affect the microbiome. And now I want to turn it around and close the loop and show you that the microbiome can actually affect the health status of the host. And this is all done with a really wonderful collaborator, Susan Erdman in MIT's Department of Comparative Medicine. And we got inspired by the study that came out in the New England Journal of Medicine, I think it was 2010, from Harvard School of Public Health, where they looked at all the different dietary and lifestyle factors that affect why we tend to gain weight as we age. Okay, so they actually went through 1.6 million years of patient data. Okay, so that's a cohort of hundreds of thousands of patients over decades to really get at what are the different factors that influence this. Turns out exercise doesn't matter. Okay, that may come as good or bad news to some of you. But diet was very important. And of course, the worst things that you could do if you're trying to maintain a slim body mass is potato chips, French fries, processed meats, and sugary drinks. Okay, so the fast food diet, we're calling it. None of this was a huge surprise. The most energy dense foods tend to be bad for you. But what was a surprise was yogurt. So yogurt was typically, across hundreds of thousands of patients, the yogurts that people are eating are very sugary. They're very energy-rich foods. They have exactly the phenotype of all of the other foods that were causing people to gain weight. But these yogurts, after you accounted for all of their other calories, the yogurts were actually making them lose weight, even though they're probably very sugary yogurts. So they said, what's going on? We can't really explain this part of the data. Maybe it has something to do with the microbiome, right? And specifically the cultures that are in the yogurt. So we wanted to see if that works in mice. These guys are litter mates, actually. These guys are brothers, okay? This one, so we initially tried it with yogurt, but all the experiments that I'm going to show you are just purified Lactobacillus reuteri in the drinking water of these different mice, okay? And so this guy had Lactobacillus reuteri in his drinking water. This one did not. And you can see, in addition to the huge disparity in weight, and I should say these are normal mice. Okay, these aren't some weird genetic mouse that tends to put on weight or anything like that. We've done this in many different mouse backgrounds, and this is very, very reproducible. So we notice a few things. One, the difference in body mass, and then two, the difference in their coats. This guy's coat is sort of disheveled, and this one is very shiny. So we wanted to get to the bottom of this and figure out what is it about the probiotic that's actually influencing weight. So the first thing that we thought is maybe the Lactobacillus reuteri is doing something metabolically in the gut. Okay? So we sequenced and we found that these L. reuteri are present at one count in maybe 100,000, something like that. They're present at like a very, very low abundance. So we sort of dismissed the idea that there's enough of them to actually do anything meaningful metabolically in the gut in terms of just the sheer volume of nutrients that the host is getting from food. So then we thought maybe what the lactobacilli are doing are they're altering the microbial community. They're interacting with all the other species and really changing the whole microbiome in a way that does have an impact on the metabolism of the host. So we looked at that, and so what I'm showing you here, each of these points is a different mouse. And it's plotted according to its the composition, the species that are in its gut. And on the left, I'm coloring based on diet. So we had half of these mice on a fast food diet, okay, the new Western diet, and half of these mice on a standard mouse chow diet. And so you can discriminate. If you look at the bacteria, you can see the microbial community is shifting with respect to diet, okay? But when we color by probiotic, okay, there's, this is the best classifier we have, okay, splitting right here, and that's, that's certainly not significant by any means. And of course, we've done more sophisticated techniques to try to see if there's anything significant with respect to probiotic, and there's really no effect of the probiotic that we can discern, okay. So if the lactobacillus themselves aren't doing anything metabolically, and they're not doing anything by interacting with the other species in the gut, then how is it that they're influencing the host physiology? And so it turns out when certainly the next thing we looked at is what are they doing with respect to the immune system, okay? So here's our 4 groups of mice. We have our control mice on an ad libitum diet. Our fast food mice on the New Western diet, okay, also all these mice are ad libitum diets. So this is our control diet, our New Western diet, our fast food diet. And here's our controls with Lactobacillus reuteri in their drinking water. And these are our fast food mice with Lactobacillus reuteri in their drinking water. What you can see is in the fast food mice, when we look at their FOXP3 positive, so their anti-inflammatory regulatory T cells, okay, in the mesenteric lymph node, we see a huge depletion of those cells, and that's consistent with generally with what other folks have seen in terms of increase in IL-17 and in fatty tissue, things like that. But when we put them back in the probiotics, you can see this huge rebound and this huge increase in the level of regulatory T cells, so these anti-inflammatory cells. And so, but I think that so we followed this up with a number of experiments. If we inject these mice with anti-IL-17 antibodies to sort of capture that anti-inflammatory effect, we can recapitulate this probiotic effect of weight loss in our control mice. If we block the pathway with anti-CD25 antibodies, then it abrogates the effect of the probiotic. Okay? If we do it in IL-10 deficient mice, we get no effect. But I think the most striking experiment is if we take a mouse and we expose it to the probiotic, and then we harvest the lymphocytes from that mouse and put them into a RAG deficient mouse that can't make its own lymphocytes, that mouse will get the probiotic benefit, whereas a control mouse where we harvest lymphocytes from a mouse that didn't see the probiotic and put it into that control mouse, that one doesn't get any probiotic benefit. So we can completely confer all of the probiotic effects by taking the lymphocytes from any mouse that's seen the probiotic and transplanting them in a RAG deficient mouse. So we really feel strongly now that we can say that we don't know exactly what the mechanism is, but it's definitely mediated through the adaptive immune system. Okay, so I think I've hit all my key points now. So microbiome reflects health status of the host. The things we do can alter the microbiome, right? And the microbiome, of course, can in turn affect the health status of the host. Now I want to revisit one of those points, which is the things we do can affect the microbiome. And so we got inspired by this paper. We're calling it the sushi gene paper. And what these folks noticed is they were looking at enzymes that degrade algal polysaccharides. Because you know, a lot of the Earth's carbon is stored in these macroalgae that have really weird sulfonated polysaccharides that are very difficult to degrade. They're not difficult in the sense that lignocellulose is difficult. Okay, if you get the right enzymes, they'll just melt away. Okay, so if you can find the enzymes that degrade them, then you have a really good alternative source for biofuel that's much easier to process than lignocellulose. The only problem is we don't know what all these enzymes are because they're very peculiar. Okay, so they were studying these enzymes, and they had solved a crystal structure for one of them. And they decided to, you know, blast the sequence databases to see, you know, what homologs for this enzyme were out there. And what they found is, well, they had the enzyme, of course, occurs in these brown algae that are used to make these nori sushi wrappers. Okay, so same polysaccharide, and so it occurs in that algae. It also occurs in marine bacteria. And the copy in the marine bacteria is so similar to the one in the algae, we can infer that the bacteria actually stole the gene from the algae, and now they live on the algae, so now they use that gene to actually eat the algae. Okay, so they stole this gene, and now they use it to live off the algae. Well, they continued to look for sequence homologs, and they found 4 homologs to this gene that were from humans. Okay? And in fact, all 4 were from Japanese people. And they said, well, this is very interesting. They eat a lot of exactly this kind of algae. And when they looked at the tree, it was very clear that the gene went from the algae to the marine bacteria that eat the algae, and then from those marine bacteria to the bacteria that are living in the guts of these Japanese people. Okay, so this was pretty. This got them pretty interested, and they said, well, you know, maybe it's just a fluke that it happened to. These 4 examples happen to be in the Japanese people, so they went to a database of metagenomic sequence from Japanese populations, and they found the gene was ubiquitous. Okay, it was very, very common, and they went to an even larger database of North Americans, and they didn't find a single copy. Okay, so the story, which is really, really cool, is that you know, there's this gene that's used to make these polysaccharides was stolen by the marine bacteria. So the marine bacteria could eat it. Somebody then harvested that whole system, okay, ate it, and then the bacteria in their guts stole the gene again from the bacteria who stole it from the algae. And now it's and then it thrived in that population, and now it's ubiquitous. Okay, and I thought this was just the coolest story ever. And I told my students, Mark and Chris here, at the time, you know, why don't you go to the sequence databases, look up all the existing genomes, blast all the genomes against all the other genomes, and find like if you could find 5 or 10 examples of something like this, that would be fantastic. You know, it would be a great paper. So the project was to find genes that are basically 100% identical, but in 2 totally different species, and those would be genes that have very recently transferred from one species to another. Yeah. And so they came back a week later, and they're like, Eric, we did what you said, and we've got 80,000 examples. So I said, you know, never trust a rotation student, so I enlisted Jonathan over here as a very senior graduate student. Didn't tell him anything about what these other kids had done. I said, Jonathan, go to the database and blast all the genomes against each other and look for these, and he came back with the same results, and then we started to believe it. And so what was even more striking is that when we looked at the origin of these genes, it turns out that almost all of those genes that were being transferred came from bacteria that live within the human body. Okay? So there were about as many environmental bacteria, non-human associated bacteria in the database at that time as there were human associated bacteria. But all of the cases where 2 bacteria shared an identical gene, almost all of them were from 2 bacteria that were both isolated from the human body. Okay. And if we use this human-associated versus not, which is arguably a very simplistic definition of ecology, okay, but one that's close at hand, we can compare ecology to phylogeny in terms of which is a stronger driver of horizontal gene transfer, okay, this gene exchange between different species. What we see is that when we look at bugs that were both isolated from the human body, this blue curve, there is a strong signal relative to distance. Okay, so more distantly related bacteria tend to exchange genes less often, but I want to point out that the most distantly related bacteria, if they both come from the human body, are more likely to share a gene than the most closely related species that we looked at, where one was from the human body and one was not. Okay, so I think we can confidently say that ecology is a stronger factor, stronger driver of gene exchange than phylogeny or relatedness is. Okay, so we use these data to make the first ever map of where genes are being exchanged between species. And so, like I said, so these are all of the human sort of environments, and now I've broken it down by specifically the body compartment that the bacteria came from. And you can see over here there's a lot of gene exchange happening, and there's less exchange between the human and these other environments. Okay, but what I'll point out is that no matter where we look, gene exchange is always stronger within a category than between categories. Okay, so even even these non-human associated environments, there's still more gene exchange within, let's say, a hydrothermal vent than there is between a hydrothermal vent and any other environment. Okay. So then we became interested in looking at what different classes of genes are doing. And in particular, we want to know how easily are antibiotic resistance genes being spread from one bacteria to another. That's an important question. And so here what I'm showing you, this is the same map. So this shows how much gene exchange there is. And over here I'm showing you what fraction of those gene exchange events involve at least one gene that's known to confer antibiotic resistance. Okay? And I want to draw your attention over here. So I said there's not as much exchange between human-associated bacteria and environmental bacteria, with two exceptions. So there is a significant amount of exchange with the bacteria in our food and bacteria that live mainly in livestock on our farms. Okay? And if we look in the other corner of this plot and we look at those gene exchanges between the bacteria in our farm animals and the bacteria in our food and the bacteria in our bodies, we find that more than 60% of all of those gene exchange events include at least one antibiotic resistance gene. And what this tells us is that we really shouldn't listen to arguments that say, oh, the antibiotic resistant bacteria that we're promoting in these livestock populations, these are not the bacteria that actually thrive in the human gut, so it's totally safe. Okay? What we're showing here is that those bacteria that live in livestock guts can actually transfer their genes to the bacteria that do thrive in human guts. And so these genes are jumping across species boundaries. And that's why we need a really consistent international policy on subtherapeutic antibiotic use from my perspective. Okay. So initially I showed you that the types of species that are in our guts are pretty stable over time. Okay. But what if all of these bacteria can pick up new genes over time? So even though the species were the same, if species can pick up new genes on a time scale that is consistent with a human lifetime, then even though you had the same species you did 5 years ago, those species might have all new abilities. Like maybe if you eat sushi every day, this would be a good experiment to do. Maybe you can get a research grant to pay for it. Eat sushi every day, in 5 years, will your bacteria pick up the genes that they need to degrade those nori sushi wrappers? Maybe, right? It depends what the time scale is. And with the assay that I've shown you, all we can say is that if the DNA is 100% identical, there could have been a transfer yesterday of that gene or a transfer 5,000 years ago. Okay, that's about the resolution that we have. And so that's not a lot of resolution to tell if it's, it can happen on a human lifetime timescale. But we started a project in Fiji, and what we've been doing there is instead of getting a metagenomic sequence or just getting molecular barcode 16 SIDs, we've been doing single cell genomes. So we can get entire genomes from single cells and get bacteria that basically come from the same person. And what we wanted to ask is, do bacteria from the same person have an excess of horizontal gene transfer? Okay? And what we find is that it looks like there is a slight excess, but the real excess is when we compare the genes that are being shared within these small Fijian farming villages to the level of gene exchange that we see in westernized countries. Then the results are pretty striking. So we can see that these genes, they might settle in individual people. There might be person-specific genes, but there's definitely sort of village-specific gene effects.
That's a good question.
Yeah, absolutely.
Go back one slide, and you have the comparison between the Fiji and the HMP. I'm wondering if how that graph would change if you had geographic information on the HMP centers, right? So
yeah,
so
so
people like living in Seattle or
right. Right.
Would you see sort of, because you're comparing like these little villages to like a sort of a more global, geographically disparate population.
Right. Yeah. Let me repeat it for the folks who are listening. And so the question is, if we look at the geographic signal within the HMP, is there an association between geography and the rate of gene exchange? So we've done that, and that's actually in our 2011 paper. And it turns out that there's absolutely no correlation between geography and gene exchange within the HMP. And I think at the end of the day, that's not very surprising, because now we just get on planes, we fly places, things like that. Whereas these Fijian villagers, it's very different. They never get on planes. In fact, most of them don't even go to the other 4 villages that we sampled. So
they share diet much more often too.
They share diet, yeah.
All that stuff, which
Yeah.
Western cultures we don't. Right?
Yes, exactly. Here's so we can look at what these genes are, and we can see that if we look at the Fiji-specific genes, our Fijian population, many of them have a very large number of the total set of genes, whereas the HMP folks generally don't have very many of the genes we identified as being transferred within the Fiji group. And of course, it's flipped when we look at the genes that are being transferred within the HMP group, the westernized population. The Fijians tend not to have them, and the Westerners tend to have most of those genes. Okay, so I should just ask, I put a ton of slides in here because they said we had no limits whatsoever, but does anyone know what time it is? I don't want to...
It's a little after 10. It's 10:07.
Oh, okay, all right. So what I want to do now is sort of take a step back, and we've done a lot of things, and we've come at the human microbiome from the perspective of an ecologist, and now I want to sort of flip it around and say, well, we have all this really interesting data on the human microbiome. One thing we have in droves is just raw data, and that's fairly that's not a situation that most ecologists find themselves in, okay? So collecting lots and lots of species abundance data in an ecosystem is a very big deal, right? And it takes quite a long time. But for us in microbial ecology, it's like, oh, you get one sample, sequence 1 million species IDs from it, and you're done. So we wanted to go back and really test some basic fundamental ecological, theoretical ecology questions in light of all of this new data that we have. And this is work done by Chris Smiley, a really terrific computational and systems biology graduate student. And so Chris was interested in this diversity stability hypothesis. This is the hypothesis that things become more stable or less stable as you increase the biodiversity in an ecosystem. And so this concept has a healthy history of debate back and forth, and sort of maybe culminating with some nice work by Sir Robert May, showing that as you add more of these links, positive and negative interactions between species, the whole mathematical system becomes much less stable. Okay? So that would sort of argue against it. And of course, most of the original arguments were sort of for it. I should point out that a lot of the debate in this field is by people using different definitions of stability and diversity. Okay, so I'm going to show you exactly what my definitions are, and I will make the point that I think so. Huh, I hope so. Hope that slide's not missing. Okay, let's wait, and if it is, then I'll just tell you what they are. Okay. But even within my narrowly defined definitions of stability and diversity, there's still a lot of debate within the literature about whether the association is positive or negative, or whether there's no association at all.
Can you go back a second and just explain a little bit about what's the difference between community stability and population stability? What are
Yes.
they actually trying to get at?
Okay, so So I won't go too far into this because this is I just pointed out for folks who know this side of the debate, and this side of the debate is was really between different definitions of stability. So community stability is something like if I look at the average biomass output of a plot of land over 1 year to the next, the higher biodiversity that is in that plot of land, the lower the variance is going to be from year to year. And that sort of, you know, that you can make a very elegant mathematical argument to explain why that's true, because maybe, you know, you get more moisture or you get less moisture, and some of the plants are you know, impacted positively by more moisture, some are impacted negatively, and the more plants you average that randomness over, the smaller your variance is going to look. Okay, so that is certainly true. That's not the kind of stability that we're talking about. What I'm talking about is if I have an ecosystem, it's made up of species A, B, C, D, and E, and A is present at 50%, and B is present at 30%, and C is present at 10%. And I look at those numbers from 1 year to the next. As I look at each of those species, if A is 50% in year 1, and it's 45% in year 2, that's a pretty stable. From the population standpoint, that's a pretty stable community. If A is 50% in year 1 and it drops to 2% in year 2, even if the total biomass coming out of that ecosystem is the same, I'm going to say on the population level that ecosystem is not very stable. So that's the kind of stability that I'm going to look at here. So I think I made this point, but here it is with a nice picture. This is Barrow, Colorado Island. This is, I think, the only macroecology data set that is comparable to the data that I'll show you from the human microbiome. And this sucker, they unleash an army of investigators and postdocs and students and undergrads and everybody that they can to divide this island up into these quadrats. And then I think they go and they count every plant that's at least 3 centimeters tall or something like that. Okay, and it takes decades to complete this survey. Okay, we can do experiments like this. Okay, so a little bit of dental plaque, and we have a whole microbial ecosystem. We can do all of the counts very accurately just by sequencing, and of course we can go to hundreds of different people and go to exactly the same type of body compartment, and then look at how the same ecosystem in a very similar environment has been assembled independently. So it's really sort of a unique data set to test these fundamental ecology questions. Aha, here it is. Here's my definitions. So I told you a little bit conceptually what I mean by stability. Diversity, I'm going to define 2 types of diversity. One is alpha diversity, and we use an entropy-like measure, the Shannon diversity. And this is basically how many different species are there in my sample. Okay? I'll use beta diversity, and that is if I look at a particular sample, how different is it than every other sample that I've looked at? So if something is really an outlier, it's high in beta diversity. It may not be high in alpha diversity, okay, because that's just how many species are there. And then stability, what I'm going to do is I'm going to say, I'm going to look at it at time point 1, I'm going to look at it again at time point 2, and 1 minus the distance between those 2 communities, that's my stability. Okay, so if it hasn't changed at all, the distance is 0, then the stability is 1. Yeah. And for distance, I'm using a metric, the Jensen-Shannon divergence. Okay. Okay, and so we went back and so the first thing we did is we looked at all these different body parts. Okay, these are data from the Human Microbiome Project, and we said, what's the stability versus the alpha, versus how many different species there are. So this is the alpha diversity, this is the stability, and we see that there's this negative correlation. So the higher the diversity, the lower the stability. generally consistent to what had been seen before, although people see different relationships. I would say most are negative, some are positive, and some people get no result. So then what we looked at is we looked at beta diversity. This is not something that people traditionally think of in terms of the diversity stability debate, but we looked at it and it turned out to be an even more significant result. Okay? But now what I'm doing here is I'm comparing all of the different body sites to each other. And so there's something funny about this because it's like comparing the gut to the, to behind my ear, to saliva. It's kind of weird. It's like comparing deserts to rainforests to temperate forests, and it doesn't, it's not exactly what we're thinking of. So then what we wanted to do is we wanted to go into like each individual body site, okay? And looking only within a body site, and here are all the sites we looked at. We looked at two things: whether diversity was positively correlated with stability, negatively correlated, or zero. And so we have alpha diversity as the first column. Blue means it's a negative correlation. Red means it's a positive correlation. White means it's a zero correlation. Okay, so some of these are empty. Okay. And what we saw is that we saw a mix, okay, when we looked at alpha diversity. Some were strongly negative, but some, like subgingival plaque here, were strongly positive, okay? And some, like the keratinized gingiva, were basically uncorrelated. And then we looked at beta diversity. And if you look down at beta diversity, every single site that we looked at was negatively correlated with respect to beta diversity. So beta diversity and stability are always negatively correlated in every site we looked at. But this is again comparing across different people. And what we really want to do is compare one ecosystem over time, right? And so we had a few data sets where we could do that because we had time series. So now I'm showing you, these are all individuals. So this is a tongue, male and female. This is palm, male, female, and gut, male, female. And when you look at alpha diversity, you can see that sometimes it's negatively correlated, sometimes it's positively correlated. So the male here is negative, female is positive, okay? And their guts really can't be that different, one would think. But when you look at beta diversity, it's always negative. So the beta diversity seems to be a more robust correlate of stability. And this was the one that I think is really kind of shocking, and that is in one of the individuals we had a course of antibiotics. So we had a time series up until antibiotics were taken, and then a time series after. And what you can see is that pre-antibiotics, the alpha diversity is negatively correlated with stability. Post-antibiotics, in exactly the same person, the alpha diversity is positively correlated with stability, okay? So the alpha diversity versus stability is not even consistent within a particular body site. It's not even consistent within a particular person to external perturbations. So the idea that there's some fundamental ecological relationship between alpha diversity and stability, we're rejecting that idea. However, again, even pre and post antibiotic, we still see this negative correlation between beta diversity and stability. Okay, and here are all the data together, and I've also added in that tropical forest on Barrow, Colorado island that I showed you. Okay, and so we see a huge range of positive and negative correlations for alpha diversity. Every single relationship, including the tropical forest, is negative with respect to beta diversity. Okay. So why would beta diversity correlate and correlate in a negative fashion with stability? Well, the way I like to think about it is this. If we have a low beta diversity, it means it's not an outlier. It's right here. If there's a stable point of the system that everything is being attracted to, then if you have low beta diversity, you're very close to that stable point. So when I come back and I look at this system again in a month or in a year, it's probably still going to be here, down here, near this stable point. Okay. However, if I'm looking at a system that has high beta diversity, it means it's definitely an outlier. It's probably very far from the stable point. And when I come in back and I look at it a year later, it's probably going to be a lot closer to that stable point because, well, it's a stable point, right? And so if I have high beta diversity, I'm going to expect to see low stability because the next time I see it, it's going to have moved quite far. Whereas if I have low beta diversity, I'll expect to see high stability. So it actually makes a lot of intuitive sense that you would have a negative relationship between these variables.
It's kind of like microbial regression of the mean, in a sense. I mean, if you think of it from a statistical perspective, right? I mean, you see outliers, you see bizarre, points of far away, and then if you see them again, you see this regression of the mean phenomenon. That's kind of like what you're describing in a way.
Yeah, so I should just be clear. I'm not offering any sort of proof that this is true. I'm giving you an intuitive basis for where we're coming from in thinking that we might expect to see a negative correlation with stability. So it does suggest there is some type of phenomenon like that. Although I think it's kind of interesting that things are. So I showed you that the time series data in the beginning, and I said, at the end of this time series, it really still looks like you're being drawn from the same random distribution. Everything is stationary. All of the driving forces that we know about in ecology and evolution, well, I should say most. Okay, most of the ones that are plausibly happening in the gut, they don't lead to stationary distributions over time. Okay, genetic drift is not stationary; it's a random walk. Natural selection is a directed random walk, so it's very non-stationary. And all these things that we know are at some level driving these interactions lead to non-stationary distributions. So the fact that I'm saying that, there is a stable point that everything's attracted to is weird. It's weird, okay? But it's supported by the first study we did. And if it is true, then all of this seems to make a lot of intuitive sense. So let me just be clear. This is by no means a proof. This is an unusual observation, and I think this is actually the main takeaway from this project. But what I want to do now is I want to go back, and now that you have an intuitive sense for why beta diversity could be negatively correlated, let's figure out what the heck is going wrong with alpha diversity. Okay. So the thing that's going wrong with alpha diversity, I think, is the way that we think about alpha diversity. And we usually think of a number as a number that exists in a Euclidean space, and there's no particular constraints on it. But in fact, let's look at a really simple ecosystem that has 3 species, A, B, and C. Okay. The possible ecosystems that you can construct from these 3 species all lie within this triangular region that I can call the simplex. So at the vertices of the simplex, I either have an ecosystem that is entirely species A, entirely species B, or entirely species C. And right in the middle of the simplex, that would be an ecosystem that had an equal amount of A, B, and C. Okay? And the only points that are valid are the points that are within this simplex. Okay? And when we measure these ecosystems, microbial ecosystems, we don't actually measure how many bacteria there are. We measure what percent is A, what percent is B, what percent is C. Of course, it's more than 3 species, right? It's more like 300, but that's the general idea. And things behave a little bit weird when you take them out of Euclidean space and you put them into and you put them on a simplex. Okay? And here's an example of how things behave oddly. So let's imagine that we have a stable point in the system, and the stable point is right here in the middle of the simplex. Okay? So the point where alpha diversity is highest, where you have an equal amount of species A, B, and C. Well, if my stable point is here, then as I deviate from my stable point, I'm deviating not only from the stable point, that's increasing my beta diversity, I'm also deviating from the point of highest alpha diversity. So then, as I deviate from this stable point, beta diversity goes up, and alpha diversity goes down. Okay? And stability is also going to go down because I'm deviating from my stable point. Okay? So there, I would expect, because alpha diversity is going down and stability is going down, I expect a positive correlation between beta diversity and stability. Now let's imagine that my stable point is right here at the vertex, where I'm dominated by species B. Well then, as I deviate from the stable point, which is dominated by this species B, my stability goes down because I'm deviating from the stable point, but actually my stable point is at the point of lowest diversity. So most directions that I deviate will actually increase my alpha diversity, okay? So increasing alpha diversity lowers stability if the stable point is near the vertex, and I get a negative correlation between diversity and stability. So now you could imagine, well, if all of this is true and all of this is absolutely true, it's just math, then I should be able to, and I believe that I can predict the stable point of a system by looking at sort of the average over all the 300 people that I have samples from. And I just look at that average and I say, this is the stable point. Then I should be able to predict whether the alpha diversity that I see is going to be positively or negatively correlated with stability. And so here I'm showing you the actual data, alpha and beta diversity correlations, and here I'm showing you the simulations, okay? And this is just another representation. This is the observed correlation between alpha diversity and stability, and you can see it goes from negative to 0 to positive. And then this is what I predict based on theory. So I would say most of the debate that might be in the literature on population level stability versus diversity, we can explain simply as a mathematical artifact of your data existing on a simplex and not in a Euclidean space. Okay. However, the more interesting thing, from my perspective, is the assumption that went into this model that seems to have very good predictive power, and that is that there is a stable point and that beta diversity is negatively correlated with stability. So what's really interesting now as a biologist is go back and figure out why the heck is there a stable point, right? Is the immune system enforcing some rules about how abundant different species can get? Are they occupying different niches that are constant based on our diets, or what exactly is going on? Okay, so I want to end up with how we're really starting to engineer the microbiome now. And I want to talk about a study. It was done by analysis done by Mark Smith and Chris Smiley in my lab. Dirk Jeevers really coordinated this whole effort through the Broad Institute. And on the clinical side, Libby Homan, Elon Youngster, and Jenny Sauk over at Mass General did all of the clinical work. And so here we're looking at C. difficile infections. Okay, the most common hospital-acquired infection. Oftentimes, so these infections crop up when you go into the hospital for some procedure. You're given antibiotics, and once you've wiped clean that healthy, stable ecosystem, C. difficile can crop up and really take over and then exclude all of the healthy bugs. We treat with antibiotics. Most of the time, people get better. But as soon as they fail antibiotics, it's a losing battle, and they're going to keep, generally, they're going to keep failing and failing and failing. And if you've failed antibiotics 3 times, then it's very likely you're just going to be on antibiotics indefinitely. Okay, so when I say fail antibiotics, C. difficile definitely responds to antibiotics, but as soon as you remove the antibiotics, it comes right back. Okay, and living on vancomycin is not fun. All right. So, or cheap. So what folks have done is they've sort of broken this cycle by pulling patients off of vancomycin or whatever antibiotics they're taking, getting healthy bacteria from a healthy donor, transplanting that into the gut as a fecal transplant, and then returning to this healthy microbiome. And I would say, so in our hands, the success rate is about 95% now. So this is a very, very effective therapy. Okay, this is just a graph showing. So this is the size of the problem. It's about this is a couple years old. There's estimated to be about 16,000 deaths this year from C. diff, It affects about 500,000 people, most of whom get better after one antibiotic treatment. If you fail once, then you see these squares keep getting closer and closer to each other. So once you failed 3 times, it's unlikely that you're going to be okay the 4th time.
So are these people who are taking antibiotics for the C. diff infection, or are they just taking antibiotics for
so the first time they take antibiotics for C. diff, then they get C. diff, and then they're put on a course of antibiotics. And so by the time they get to us, they've failed at least 3 times. And so yeah, at least they've been on antibiotics for C. diff 3 times. Yeah. Okay. But you know, there is a cure. Okay. What we know about it now is that it works. We don't know much about the mechanism. Okay. So Libby wanted to start just a randomized control pilot study. Okay, so she recruited 3 donors and 20 recipients. Okay, so this is done by Libby Homan, Elin Youngster, and Jenny Sauk over at MGH. Dirk Jeevers and Bruce Byrne at the Broad Institute collaborated on the sequencing side, and then Chris Smiley and Mark Smith in my lab did analysis on these data. And what you can see is so here are the different donors. Okay, in green here, so donor 1, donor 2, donor 3. Here are our recipients before FMT, and you can see that over time the recipients are each moving toward the donors. Okay, so their microbiomes are shifting to a healthier microbiome. Okay, and when we look, we can identify. So here's our recipient. Most of the community that we can identify the origin of is from the recipient. Okay, and these bugs go down over time. The strains that we can identify from the donor are going up over time, and then there's some number of undetermined strains. And what we wanted to do is, this is all based on 16S data, we wanted to go into metagenomic data so that we could resolve the difference between one strain and another at the strain level rather than just the species level. So this was the pipeline. We got samples basically before and after fecal transplant of stool from the recipients, and we also have samples from the donors, so we know what bacteria are going into the patients. We get the raw metagenomic reads, and then those reads we basically recruit onto reference genomes. So now we have about at least 1,000 reference genomes of bacteria that tend to occur in the human gut. So we can take these short reads and just map them onto these complete genomes. Okay, and once we have them on the complete genomes, then we can identify where the SNPs are, and we've developed an algorithm that allows us to put all of these metagenomic data in. And remember, these are very short reads, and then infer what the entire genome looks like. So we take the reads, map them against a reference, and then we break them into, let's say, 10 or 12 different strains, we call them. And so then if we have a particular species, we'll call this species 1. Here's 4 different samples. And so these are all the same species, but now I can show you what is the abundance of each of the different strains in that species. And that's based on the single nucleotide polymorphisms that align to these genomes. So here are 2 examples. So here's a Ruminococcus species, okay? And this is what's in the donor, okay? Donor number 1 and donor number 2. So donor number 1 has 2 different strains of Ruminococcus, and donor number 2 is 4 different strains. Whoops. Oh, that's unfortunate. Oh, I can't even fix it because this is just a PDF. But had I actually had the data, what you would see is that in this case with Ruminococcus, we took 4 more samples from these patients, and the patients basically looked exactly like the donors. On the other hand, when we started with Faecalibacterium prausnitzii, the patient on the first time point looked very much like the donor, like the stool that we had put in, but then an entirely new strain came up that we hadn't seen before in either the donor or the patient. And so what we're working on now is more sensitive methods to figure out where did this new strain come from? Was it present at very low abundance in the donor and it just was more competitive in this new environment? Or was it present at very low abundance in the patient? Or perhaps it was just inoculated through the food or through the outside somehow. The other thing I'll point out is that these have surprisingly long-term effects. So here I'm showing you weeks after fecal transplant. So this is least similar to where the patient started. This is most similar. And so 1 week after, they're already pretty different, and they continue to diverge from their starting condition, which is good because their starting condition was very sick over time. But do they resemble their donor, and if so, how? So one hypothesis is that they resemble their donor because they're literally just pulling those strains in from the donor. And an alternative but very plausible hypothesis is that once a little bit of the donor material got in there, they're actually, it was enough to kickstart their own microbiome and just reform a healthy community. It might not be exactly the strains from the donor. It just looks, it looks more like the donor because the donor's healthy and the patient is healthy, okay? And it looks less like a dysbiosis. So to measure that, we looked at the similarity to donor, okay? And this is pretty constant. It maybe spikes up at week 4, but when we compare it to the similarity of another healthy donor, even after 4 weeks, it's much more similar to, each patient is more similar to their own donor than to another healthy donor. So we reject the hypothesis that it's similar to the donor just because the patient's getting healthy. No, it's actually similar to that specific donor and not just any healthy person. All right, so C. difficile is it's a big problem, but it's a problem that we can solve, okay? The one problem is that fecal transplants are very effective, but they're not very available, okay? And another problem is that the FDA regulation in this area is quite uncertain. And we just wrote a opinion piece in Nature a couple of, maybe a month ago on how we think that the FDA should regulate fecal transplants. So to address the sort of both of these questions, we started a nonprofit organization called Open-Biome, and this is the first national and I think the first international stool bank. This slide is very much out of date. We're currently partnering with, I think the current number is 45 hospital networks to provide stool samples to doctors who want to perform this procedure. And we've treated, I think, almost 500 patients so far, and the growth is really exponential here, with about a 94% cure rate. And we're aiming to really treat tens of thousands of patients in the next year. And really our mission is twofold. One is to provide safe access to this treatment for C. difficile, but then also to move into new conditions where fecal transplantation might be useful. So I should note that fecal transplants were described in Chinese traditional medicine over 1,000 years ago. They've been used in veterinary medicine for a very long time. And so we're pretty optimistic that there's going to be a lot of other diseases that might respond to fecal transplants besides these C. difficile infections. Yeah.
Do you have data that of these people, of any of these people before infection? So you can kind of show that, okay, you do this, they have their normal stable point, and then of course you do this transplant.
Yeah.
They adapt or they take on some characteristics of the donor, but over time they sort of return to their normal
Yeah, yeah, yeah. No, I think so. So the question is, do we have data not pre-transplant, but pre-sickness?
Yeah.
And unfortunately, we don't. I think that would be fantastic data to get for exactly the reason you're talking about. It's probably an experiment that we can do in mice, maybe. And as we move into some other conditions, so people are interested in fecal transplants for MS or for autism or all of these conditions where the microbiome might look healthy to begin with, then maybe we can compile some of those data.
So you mentioned mice. That kind of occurred to me when you were going through this.
Yeah.
Are they a reasonable model to work out, for example, maybe an organic fecal transplant isn't absolutely essential, but maybe there are a few organisms that can do the heavy duty to displace C. difficile and let.
So there is a good mouse model for C. diff. And then for other indications, mouse models are
and does this work?
Good or bad.
Does a fecal transplant from a mouse infected with C. diff?
Yes, yeah. Yeah, there's good paper by Lawley et al., I think in the last year or something, maybe, I'm going to say like Nature Medicine or something like that. Very, very good paper sort of establishing the mouse model and showing the efficacy of fecal transplants. For other conditions, everybody's interested in inflammatory bowel disease. The mouse models, I think, are not very good. But we're looking at other animal models. So apparently dogs get IBD. They
tried this in humans that have IBD or especially IBS?
Well, so this is where it comes down to, this is part of our mission at OpenBiome, and is really to enable research in this area. So this is a nonprofit organization started by Mark Smith, who's a graduate student in my lab. And the lab is collaborating on the sort of research arm of this. So we're partnering with a lot of clinicians to do human trial that want to do human trials for FMT by providing them with ready-made product, and then in return we're trying to collect a lot of the data so we can do sequencing and analysis on the backside. So the only problem with doing this, at least in the U.S. right now, is the FDA regs. So I'll show you a study that we started, Mark started with Athos Busvaros over at Children's Hospital. And this was prior to the new FDA regulations. And so what we did is Athos recruited IBD patients. And here's the microbiome. These are just each one of these lines represents a different species, and you can see that pre-FMT the diversity is quite low. Okay, this is the donor. Okay, here's the patient 3 days after FMT, and then 48 days after. And this was just a pilot study. We only looked at a few patients. There have been a few other pilot studies for IBD. And the results are currently mixed. There's usually a remission of symptoms early, and then symptoms come back after, let's say, 4 to 6 weeks. And what we're noticing is that, at least in our data, after about 4 weeks, the bugs are the bugs that were transplanted in are starting to disappear as well. And so we think this is actually pretty interesting. It's probably not going to be as easy as treating C. diff, but there's a lot of different varieties and variants of the standard fecal transplant that we can try. And of course, we're trying to move beyond fecal transplants to very safe and reproducible synthetic cocktails of organisms. So this is another analysis of the study that I showed you that was done by Dirk Jeevers over at the Broad. And what you're seeing here is this is one donor into 15 different recipients. Here are all the core organisms of the donor. And these 6 down here, these 7 down here actually, are bugs that, that appeared in every single recipient. So these are currently one of our targets for isolating and making into a synthetic cocktail. Like maybe, since these are the only ones that appeared in all the patients, maybe you could do a fecal transplant with just these 7 pure bacteria grown up in isolation. So that's it. Thank you guys. Thanks for letting me talk as long as I wanted. Thanks for paying attention. And if folks have some questions, I'll be happy to answer them.
Before we move on to questions, I would like to acknowledge Stephen Scott. He is our co-host, arrived early on in his presentation. We have time for a few questions before we take our coffee break. And I'm going to pass the mic around so for recording purposes. Okay, Jen, let's start back there.
So that was really wonderful. I'm a reproductive biologist, so I don't really know very much at all about the microbiome, but we're starting to think about it. And so my question is, when you do the lactobacillus experiment, you're mostly looking at the gut, but the reproductive tract has lots of bacteria as well. And so is there crosstalk between those 2 different microbial populations? And so is it that crosstalk that's important for impacts you might have, say, at the level of the ovary? Or would it be whatever's happening in the gut is affecting immunity, and then that's affecting the ovary? Or could it be both?
Yeah, no, that's a great question. Especially with respect to the probiotics, because I mean, lactobacillus. The niche of lactobacillus is really the human vagina. Well, not just human, right? So there's definitely very close relationship between the reproductive tract and these probiotics that seem to be functioning when you put them in the gut. But and we've got actually a number of papers out already on different effects of the probiotic, and even more sort of in the queue. And the take-home message is that the probiotic effects, I think the probiotic effects that most people have been searching for, okay, are effects on digestion, effects in the gut, but the probiotic effects that we're measuring, that are the strongest phenotypes, are really systemic, and to a large extent they seem to be mediated by different hormones. And so they do, I mean, testosterone is one of them, oxytocin, and obviously those have huge effects on the reproductive system. So I think the take-home message is that whatever is going on is an interaction between the bacteria and the adaptive immune system, and that somehow all of these hormones are strongly involved as well.
Okay, Ken.
I have 2 microbiology questions. The first one has to do with when you see that sudden shift when one bacterial species totally disappearing and another taking over, do you see any role of bacteriophage or viruses at that occasion? Are there?
Yeah, no, we're very interested in phage, but we're currently not. We don't have any data. We don't have any data on the phage sort of over time in the gut.
Second question: do you have any data showing the presence of fungi, yeasts, or even say methanogenic or archaea?
Yeah, so there's certainly archaea, not very common, and some of that could be due to the fact that our primers might not be as good for those, so it might tend to underestimate, their abundance or their prevalence in the population, but they're certainly there. With respect to the eukaryotes, so in our Fiji study, we explicitly looked for eukaryotes, and they're very abundant. Okay, in the Western populations, there's not a lot of data. We've gone back and looked for it in the metagenomics, and there's also there's not, let's say on a relative scale, there's a lot less eukaryotic material in the stool than let's say in the Fijians. One sad thing is almost every Fijian has plant material in their guts, and something like 30 or 40% of us have plant material. I guess we eat like super processed foods where we can't even see the DNA of what we're eating anymore. But
okay, 2 more questions. Jack 1 and Xiaozu.
Yeah. Okay. When you do the fecal transplantation, you find a new strain of bacteria.
Mm-hmm.
You said that either they are undetectable before you transplant, either in the donor or in the patient. Is it possible there are a new recombinant strain? Because you transplant the bacteria that generated a new strain, not from food, not from the donor, not from the patient. Is that possible? Because they can recombine
mm-hmm.
donors, the bacteria can recombine it with the patient's DNA.
Yeah.
They can generate it. Is it possible?
I think so. It's certainly possible. I think as often as we see this happening, I think it's definitely not going to explain that pattern that we see, because it's fairly common to have another strain come in. But I should say we haven't really gone through thoroughly to see if there's any evidence for these strains in either the donor or recipient. So I think we need to develop a more sensitive analysis first. Certainly, we can figure out if what you're saying is true. We have all the sequences, just the analysis tools that we need to tune to be a little more sensitive. But we can certainly test that hypothesis, and I think it's a good one. It would be neat to see if that happens. But I suspect that the timescale for that is that those are very rare events within any one person.
Could you comment on cross-species fecal transplant, such as we have plenty of cow and pig here, and we transplant the fecal material from those animals to human body? Is there any safety concern? Or is any trial? In that case,
well, I would never do that.
Yeah, in that case, there's no limitation of supply. We can provide a lot of those material, and also we can raise those animals specific environments which make better fecal material, which fit better treatment of human body.
It might be a hard sell to patients. Yeah. Yeah, I mean, maybe if there's some strain that you isolate from an animal that has some, particularly strong, I don't know, anti-inflammatory properties in the human body, maybe you could grow that up in pure culture and use that as some sort of probiotic, or put that into your fecal transplant. I just don't think anybody would sign up for a fecal transplant from a pig, although one of our clinical collaborators, and this is one of the proposed FDA regulations, is that people find their own donors, which is very onerous for patients and very embarrassing when they have to ask friends and family. But one woman came in asking if she could get a transplant with her dog's stool. And so I think that's one of the, I think that would be a fantastically bad idea, I think that's really one of the things we're trying to facilitate with the nonprofit with Open Biome is alleviating that burden from patients on choosing a donor. Yeah.
Let's thank our speaker one more time and